Okay, everyone. Welcome back to our final session on spatial analysis. Just a brief refresher of the ARC pipeline. Everything we're going to talk about in this session is also in ARC analysis. The first session today was all about this half, taking your cleaned, processed images and generating annotations. The second part of today, this session, is going to be what do you do with those annotations? Spatial LDA, K-means, pairwise enrichment, all of these things we'll be going over, what their uses are, when we do which ones. Overview, we'll first talk about choosing the right tool. So there's a lot of different spatial analysis tools. We're only going to cover a subset of the ones that our lab routinely uses. How do you decide which ones to do? When are they useful? What are the specific use cases? We'll then go through the three, I would say, bread and butter tools that we use. Pairwise distance analysis. This is for identifying specific population enrichments. There'll be an application example for pregnancy. Then K-means neighborhood analysis. This is about identifying neighborhoods in the data. And then we'll talk about pre-invasive cancer. And then spatial LDA, talk about why we use, what the advantages and disadvantages of that neighborhood approach are compared to K-means, and an application in breast cancer. The goal of this session really is to understand the benefits and the use cases of the many different spatial analysis tools that exist and give you a good handle on where to start and with which one. And with that, I will turn it over to Ina for our first talk.
Trying to freeze me to death. Okay, where does this go? Okay, so as Noah mentioned, we're going to go over some of the tools that we widely use in the lab. It's not an exhaustive list, but hopefully it will be very useful for you as well. All of these tools are going to be available in ARC. So where we left off at the end of the previous session was the cell phenotype map, a map that can overlay the segmentation of the cells and their assigned phenotypes from clustering. And the question is, what can we do with it next? So now we could look at the frequencies of cells. We can look at cell frequencies throughout the entire cohort or under some subsetted conditions. We can also look at the expression of functional markers we're interested in across the various cell populations that we identified. However, both of these types of analysis, they leave out the spatial context of the cells that is available for us since we did do imaging, and these can also be applied to any type of assay that uses dissociated tissue to give an approximation of the real frequencies of cells inside the images we're looking at. So we would like to incorporate the spatial context, and first we'll start with an example of how this could be very, very important. What you see here are two cell phenotype maps for two triple-negative breast cancer patients. The one on top has this really like salt and peppery mixing of immune cells in orange and tumor cells in blue, while the bottom one has compartments. Immune cells tend to be next to immune cells, tumor cells are isolated in their own compartments. And as it turned out, this is a very good predictor of patient survival, as you can see here. So spatial context is important, but how can we get to it? In the previous images, the pattern of compartmentalized versus mixed, it was very clear just by looking at the cell phenotype maps. However, that is not always the case. And also we need to keep in mind that we will be dealing with large cohorts, many different markers, and the patterns could be more intricate and delicate. So kind of manually looking for them is not really feasible. So if we want to do this at scale, we will need some computational tools to help us along the way. And today we're going to look at some of them. The first tool we're going to go over is cell-cell enrichment, which will allow us to see which cell types tend to be enriched one next to the other or preferentially avoid each other. Same algorithm can also be applied to the enrichment of cells around histological features. These can be arteries or glands or anything else you can denote inside your images. And finally, we're going to see two different algorithms to tease out cell microenvironments or recurring spatial features that involve several cell types inside the images. Okay, we'll start with like briefly going over each of these three algorithms, and we're going to get into more depth into every one of them in the subsequent talks. We'll start with pairwise enrichment. What we're trying to figure out in this example is whether the red cells are preferentially enriched around the green cells or not. So to do that, first of all, we can count how many times we see them closely interacting. But if we just stop there, we don't take into account a lot of the things that Candace mentioned in the previous session about how dense the tissue is, how many overall cells are there, how closely are they packed inside the tissue, and also how many green and red cells do we even have in that FOV. So to take all of that into consideration when determining if there is a significant enrichment of these cells, or if there isn't, we're going to have to create a null distribution, which will tell us what we would expect the number of close interactions be like by pure chance. So to do that, what we're going to do is take these red cells and start just shuffling them around and assigning them to random other cell locations, and then count again how many close interactions do we have between the red and the green cells, which will allow us to create this null distribution. We can then z-score the actual observed value of close interactions against the null distribution to figure out whether that amount of interactions was significant or it could have occurred purely by chance. Next, Candace and Adam are going to talk about two algorithms that are aimed at identifying microenvironments that involve several cell types and can be a recurring feature across your images. I'm just going to briefly mention the differences between the two and let Candace and Adam go in depth into each and every one. So KNN neighborhoods outputs an assignment of one microenvironment per cell. It's just an additional label like a cell phenotype, while the output of spatial LDA, the other algorithm, is a bit more complex. It gives you a probability map, which means that it assigns the probability of each cell belonging into each of the microenvironments. Okay, so we promised at the beginning, we're going to tell you which tool to use. So which tool to use? If you are interested in creating sort of a roadmap of interactions between the various cell types, pairwise enrichment could be the way to go, because you can assume that if two cell types are preferentially enriched around each other, perhaps they're indeed interacting. But if you're more interested in the overall structure and compartmentalization and things like that, KNN neighborhoods or spatial LDA would be more informative. And finally, I would like to give an example of how you can use the output of these microenvironment algorithms and plug it into subsequent analysis to get some more insight about what is happening in your tissue. So what you can see here are in the rows are microenvironments numbered from A to J, and the columns are different cell types. The color indicates the composition of each of the microenvironments in terms of cell types. And over here on the right side, you can see this tiny heat map that describes the expression of an interesting functional marker, in this case, galectin-9, in fibroblast cells. And you can see that the expression of galectin-9 in fibroblasts really varies across different microenvironments. And you can see this nice peak in microenvironment G, which is rich with NK2 cells. So what all of this could mean is that there is something about the interaction between fibroblasts and NK2s inside that tissue that could induce the expression of galectin-9. Okay, so just another brief comparison between spatial LDA and KNN. They do similar things. So when you'd like to choose between them and see which one you would rather use, just want to highlight some different advantages each of the methods has. So KNN is simpler. It's simpler to understand what it does, and the result is more interpretable. It will give you exactly one microenvironment for each of your cells, which can be easier to plug into the next analysis you plan to do. Spatial LDA will likely outperform the KNN option on boundaries between large microenvironments, but the output is a bit more complex. It will give you probabilities, and you're going to have to think about how to use that information with the next thing you do. And are there any questions?
Okay, let's dive in.
Everybody's scared. I see.
Yeah.
Okay, fine. So you're stuck with me for one more. Okay, so now we're going to go into a bit more detail into the pairwise enrichment algorithm and the various options you can use when you're running it and how to actually run it as a part of ARC analysis. Right, so back to our favorite image, the cell phenotype map. What we would like to do is take our cell phenotype maps for each of our FOVs, do our space analysis, and get this type of output, a heat map that indicates the z-scored spatial enrichment of each cell type against the other cell types. And instead of cell types, we can also have features. And at the end of the day, after we see which features or cell types are enriched one around the other, we can try to say that that indicates some sort of interaction inside the tissue that is functional and important, and try to get at this type of interaction map. Okay. Right. So let's get back to an example that Candace used plenty of times, the lymph node. Inside the lymph node, we have the germinal centers, which are rich in B cells, the not germinal center with a lot of T cells. This is what a MIBI image of this region would look like. You can see again the compartmentalization inside, and the cell phenotype map also highlights that. And we're going to use this example to try and figure out enrichment of cell types around each other inside the germinal center. Okay, so again, we start with the cell phenotype map, and we would like to know if the pink cells and the blue cells have a preferential enrichment around each other. So what we would do after we count how many interactions do we actually see in our image between the blue cells and the pink cells, we are going to start shuffling them around and counting interactions in each random permutation to create our null distribution. And we can do this globally, which means that when generating the null distribution, we allow the random permutation to assign these cells to any other cell in the image. But there is another option. Since we know that this tissue is kind of compartmentalized, and for instance, B cells are unlikely to be found all over this region right here, but they're much more likely to be found here, we can restrict the null distribution and allow only random permutations inside that region of the tissue or inside that context. And this is called context-dependent pairwise analysis. And here you can see that as we randomly permute these cells, they still remain inside the area of the germinal center. You need to figure out, depending on your data, what type of analysis would make sense for you. And it's not about right or wrong, it's just about what is it that you would like to know. Okay, so after we have our null distribution, we can z-score the actual observed number of close interactions against null distribution to figure out the significance of that interaction. And that is a nice feature of the pairwise cell algorithm, that it doesn't only give a result, but it also indicates the statistical significance of the result, which makes it easier to kind of figure out what would be important to look at and what is not so strong. Right, the notebook. When you start running the notebook, which is available under ARC analysis, learn from a lot of people's mistakes. First of all, please make sure that all the paths to all of your data folders are correct. This is, according to the computational team, the most common source of error and frustration that they encounter. And it's just as likely to happen to experienced users as well. So keep it in mind and organize all of your data the way it's supposed to be organized and where it's supposed to be organized to run the notebook. Okay. That is the most important thing, but there are several other issues you might want to adjust. So first of all, I was talking about close interactions. Close interaction is just a code name for the minimal distance you want to have between two cells to actually say that they are neighbors or closely interacting, and count it as one of the close interactions inside your image. And that minimal distance you can tune inside the notebook. We usually go for 100 pixels, and in our data set, this corresponds to about the distance of 4 lymphocytes. Once you've tuned that, again, paths where all the files are, you need to specify where is your post-clustering cell table that indicates the location of each and every one of your cells, their phenotype, and maybe some other features that you might want to use. When you call the function, there are a few more parameters you can set. So first of all, how many random permutations do you want to do when you generate your null distributions? Whoever wrote 5 here got the wrong answer, because we would advise to use something in the range of between 100 and 1,000. Going below 100 might be too little and not sample the null distribution accurately enough. Going above and beyond 1,000 will slow you down, and really it's not guaranteed that it will add any useful information or give you a more accurate approximation of the null distribution. Next is 2 optional parameters. The first is the context. So if you remember in the previous example, we can do the pairwise enrichment analysis with or without context dependence, and that only influences the part of the null distribution generation. So this means that when the null distribution is generated, you will only be permuting cell labels inside a certain context, which you add as an additional column to your cell table that adds the context or compartment to each and every one of your cells. And we've mentioned previously that you can use this algorithm not only for enrichment between cell types, but also for enrichment between cell types and some histological features. So for instance, if you would like to know which types of cells are enriched around arteries or glands inside your tissue, what you could do is add more columns to your cell table, which indicate the distance of each of your cells to the nearest feature of that type. And the output for each FOV, which goes in basically as a table shape of the CPM into the algorithm, would be this type of heat map, which indicates the z-scored enrichments between each pair of cell types, or histological features depend on what you fed into the algorithm. And now I'd like to show an example of how we use this to explore spiral artery remodeling during early pregnancy. We'll start with just a bit of background. So during early human pregnancy, and the placenta is created, it then attaches to the maternal tissue. And after that happens, the spiral arteries on the maternal side, which are just called spiral because of the spirally shape, they undergo a very extensive process of remodeling. So what happens is that they become dilated and they lose some of their smooth muscle layer. And the purpose of this is to supply the fetus with a high volume of blood, because this is the only source of nutrients and oxygen it has, but to keep it at low pressure so that it doesn't harm the delicate tissue of the placenta or the fetus. So this is very important, and if remodeling goes wrong, it can cause all kinds of pregnancy complications like miscarriage or preeclampsia or growth restriction of the fetus. So it really is a crucial process during the first 2 trimesters of pregnancy. So we can assume that if a specific cell type is enriched around arteries in this tissue, they're not just innocent bystanders, but active participants in the remodeling process. So we will be looking for cell types that are enriched around the spiral arteries throughout the 2 trimesters. Okay, no. We'll start with MIBI images, which already have segmentation, turn them into our cell phenotype maps, and we're going to have to annotate the features that we're interested in, the arteries. We'll then throw all of this into the pairwise enrichment notebook, which will give us for each FOV our heat map, which will indicate the z-scored pairwise enrichment between all the cell types, and also an additional column for enrichment around the arteries. And we can stop there, maybe average over our entire cohort to see overall which cell types tend to be around arteries, but why keep things simple when we can make them more complicated and add spatial dynamics into the mix. So the remodeling of spiral arteries follows this typical route of progression. We start with these unremodeled small arteries with a thick layer of smooth muscle. Then the smooth muscle starts to get disrupted and the endothelium swells. After that, we see fetal cells appearing on the scene. And after that, as they invade, the vessels become these dilated vessels that you can see here in stage 5. But this is not a discrete process, and it can be just as easily described as a continuum. And we can define a pseudotime coordinate named delta that just describes where along the way between unremodeled arteries and full remodeled arteries, each and every one of the arteries is. And we can then take our heat maps for pairwise enrichment and arrange the FOVs according to the remodeling score of the artery inside the FOV and get the cell enrichments around the arteries as a function of remodeling pseudotime. And we can then explore temporal trends in remodeling and see if there are specific cell types that are enriched around arteries during specific stages of remodeling, which might indicate that they are, again, active participants for that stage. And if we were looking back and only averaging across our cohort, these types of trends could have been easily missed, because if there is a specific cell type that is only enriched around arteries for a very specific time point, when we average over all of the time points, we just might lose the trend. Right. So the literature has plenty of indications that maternal NK cells are active participants in remodeling. In our data set, we found 4 different subtypes of NK cells. We were interested to see which NK cells are actually doing something and when do they contribute along this pseudotime of remodeling. So we get back to our heat maps, the output of the spatial enrichment analysis. Again, we can look at our column that indicates enrichment of cell types around arteries, order all of our FOVs by the remodeling pseudotime, and have the enrichment of each and every cell type as a function of remodeling throughout our cohort. And when we did that, we indeed saw a significant trend, a transient trend for NK2s. And you can see here in the linear regression version and the violin plot version, NK2s have this transient increase in enrichment around arteries around the 2nd stage of remodeling. And this is approximately when the smooth muscle layer gets disrupted. And this is a very good indication that this is the type of NK that actually participates in disrupting the smooth muscle layer during spiral artery remodeling. So just to summarize, we saw that the pairwise enrichment analysis can give you insights into which cell types prefer to be around each other. Also, if you were more interested in the very negative z-scores, which type of cells prefer to avoid each other as much as possible. The output will be per FOV. We will see for each FOV which cell types are enriched or avoiding each other. We can plug that into any of our subsequent analysis. And all of this is available for you in ARC. And maybe now somebody has questions. Going once, going twice. Oh, thank you, Brian. Yes.
Have you tried running this on a tile?
What do you mean by a tile? Okay, so you want to randomize the labels across the different FOVs?
Yeah, so essentially, instead of doing it per FOV, you would file the FOVs randomize. I mean, potentially if you have your delineated section randomized across FOVs.
I can see how it would make sense for the big tiled images that you have of the brain, but we haven't run it in this case because the different FOVs, they were taken from completely different locations. So it really didn't make sense to allow the null distribution to kind of permute between the different FOVs. But if you kind of comprise one big happy image out of your tiled smaller images and you do view them as one image, then yeah, I guess that would be, you know, a good option as well.
Like how is it like you combine it in workflow, and how would you do it? Like get another view in the part.
Yeah. So
So the question was, how do you integrate Pixie with the spatial analysis?
Okay. So the output of Pixie is directly inputted into this analysis because Pixie is where we get the phenotyping of the cells. Oh no, the pixels themselves, we don't take into account. We're just looking at overall what is the label that is associated with the cell. And it could be a cell phenotype from Pixie, but it can also be other things, like any type of label that you can put on your cells would do. It just really depends on what you're trying to figure out. Mm-hmm. So you're going to have to think about how that would fit into generating the null distribution. If you want to use the exact same way it's done here, which means just taking things and permuting them to the locations of the other labels, then you would need to kind of boil all that down to having one label per location that is permutable, and you can then use it out of the box. But if that doesn't make sense for generating the null distribution, you're going to have to make some changes.
So many more people. So actually the neighborhood houses that were come on naturally work better. Theoretically, I don't see why not. You could.
I think the, but I think even this sort of prior knowledge argument against doing that is that going in, right, that cell type and pixel clusters are correlated, right? So it's sort of, so in other words, when you're sort of artificially increasing the permutation space by letting parts of cells be randomized. You know what I'm saying?
But I wasn't talking about like the question was like what is the feature
oh, I see.
Include them into this.
Good, like ECM and yeah.
Like using the pixels that comprise these features, yeah, that would be a way to integrate them in.
Yeah, I mean, right now, I guess, you can kind of, I mean, maybe one thing you could do for stuff like collagen, sort of these extracellular matrix components, is you could maybe randomize cellular objects with respect to like ECM pixels. No, it's a good point because I think there's actually stuff that we're interested in right now that has a lot to do with like, how tumor-associated macrophages and CAPs and stuff like that, like how they're near or far away from sort of remodeled ECM. So it's actually pretty cool.
Yeah, so the discussion was, what about if you have ECM pixel clusters or things that are never going to be within cells? How would you include it? And the short answer is don't know yet, but we'd like to.
We'll think about it.
We'd like to find out the best way to incorporate it.
Summarize it somehow. And I think Candace is saying summarizing at the pixel level might be really data intensive, but if you could turn them into objects or neighborhood features, you could factor. They could be factored.
If they can be labeled, they can be permuted.
The analysis pipeline.
Yeah, and I think, and this kind of gets to almost like a philosophical question, I guess, because it's, one of the things that we've debated back and forth is it's sort of, it makes sense to look at random permutations, in most cases with cells, because cells are known to be able of being at different places in the tissue, right? But then, most of the time we treat larger objects, like for instance, a blood vessel or something like that, as being a stationary feature, right? So it's sort of, things can come to it, but it doesn't really go to things, right? And so it may be that it would make sense to sort of treat the ECM the same way, which is that, it gets deposited, it can be broken down, but it's not mobile. And so, and that kind of gets into some of the fun things to think about when you're trying to figure out the right way to ask some of these questions, is to really think about, logically, what makes sense when we're saying, let's randomize this to this, right? Is that actually recapitulating something that we
could actually possibly happen or not. And so and I think that's to Inna's point. That's why we're trying to kind of just lay out the high level concepts around these, because there's probably not hard and fast rules for all of it.
Yeah, just more of the same. Sorry.
It's so snarky.
He's slowly but surely losing it.
Why is the cell by cell type heat map not symmetric?
Because when we create the null distribution, it's a question of which cell type remains stationary and which cell type starts to move around it. You can also make it symmetric. That is also okay, and just take the two cell types and let's reshuffle both of them, and then it would be symmetric. But the way we do it, and what makes more sense to us at least, is to keep one cell type stationary, take the other one and start permuting it around, which means that it doesn't have to be symmetric.
Where does the context column come from? Is that generated automatically? Is there a manual step?
It's a manual step. Like, you need to figure out what your context is. You need to assign each and every one of your cells to a context. So it just depends on what the context is and how are you generating it. Are you identifying it automatically from your images, or are you actually manually annotating, that sort of thing?
For creating the null distribution, what if you don't know what the typical distribution of your cells are? For example, CD20 and lymph node is very well established, but what if you're looking at a tumor where you don't even know what your baseline assumption would be?
Well, I think that it can be approximated if you keep, for instance, the permutation inside the context of the tumor, because you would expect it to be different from the non-tumor tissue, and try to do the permutations. This could get like a bit difficult if it's a very rare cell population, because it would be really hard to assess what is the null distribution just out of permutations. But this is just a difficult question, like what happens with very rare cell types. So I don't know. Okay.
So the question was, how do you decide what distance limit to use? We use 100. How did you pick that?
So it was picked specifically to the tissues we're looking at. It made sense, but you can play with it. And I've actually tried to decrease and increase to see if perhaps I think that the short answer is, if you think it makes sense, run it with several thresholds, because you might pick up, when you go to a lower threshold, you might pick up on more delicate trends that you just did not see when you were looking at like a larger distance around whatever it is that you're looking at. But for the most part, it will be robust around the 100 pixels limit. Does that answer the question? Yeah. Okay.
And in it, you've brought up some ideas before about how you could maybe do something similar without having to specify.
Yeah, you can also use it's not implemented in this pipeline, but you can also think about distances themselves, like without thresholding and saying close interaction, not a close interaction, which will be in some cases sensitive to that threshold. I mean, you would hope that for the most part it will be robust and you can test around it, but for some instances, yeah, it will be sensitive to the threshold. And then you might want to look at the distribution of actual distances instead of counting just occurrences, was close, was not close, was close, was not close, but to create a null distribution of distances from that feature.
Awesome. Thank you, Ina.
Take my necklace.
Put it over here on this side.
Okay. Just put it behind me. Okay, hi everyone, I'm back to talk about K-means neighborhood analysis. So this is really like the second class of spatial analysis that we typically do in our lab, where we try to identify neighborhoods in our data. So first I'm going to go over what neighborhood analysis is, give an overview of K-means to find these neighborhoods, talk about some practical ways to apply this to your own data, and give an example in ductal carcinoma in situ or DCIS. Okay, so what is neighborhood analysis? So we use the terms neighborhood and microenvironment kind of interchangeably. Yeah, so if you hear one or the other, they're referring to the same thing. So cells are regulated in part by their local cellular niche and can be heavily affected by what cells are around it. And so we often will see collections of these cell phenotypes that are spatially co-localized and conserved across data sets. So in this example here, like if we're looking at a tissue, we can define these neighborhoods where each different neighborhood is characterized with a different frequency of the cell phenotypes that make up that neighborhood. So the point of neighborhood analysis is to try to find these different neighborhoods that are occurring in our data set. Okay, so to give an overview of neighborhood analysis, so hopefully you're used to seeing these by now, cell phenotype maps where the colors correspond to the different cell types. And so for every single cell in our image, what we do is we look around that cell for some radius around that cell and simply count the number of cells around it. And so for instance, here, these like pie charts are denoting the composition of the cells around that index cell. So we do this for every single cell in our data set. And then identify microenvironments by using those neighborhoods as the feature vector and assign each cell to a microenvironment. So now in this map, instead of each color corresponding to a cell phenotype, the colors correspond to the microenvironment or neighborhood that it was assigned to. So for example, here, like this green microenvironment, hopefully this is the same lymph node image we've been looking at this whole time. This green microenvironment is the T cell zone. If we look on the heat map, it's defined by the CD4 T cells. This like light orange one is the B cell zones that are B cells, and the germinal center is largely defined by FDCs. Yeah, so this is a very simple example of like the kind of microenvironments you could expect to see in a lymph node. So one way to do this step here of actually identifying the microenvironments is to use K-means clustering. So K-means clustering is a very simple algorithm, which is one of the big advantages of using this method. It's very interpretable. So what you do is you first initialize your cluster centers, and this is done randomly. So you can imagine that all of these gray points here are all of the cells in your data set, and you just embed them into your n-dimensional space. In this case, it's just 2 dimensionals for easy visualization. And then you initialize your cluster centers randomly. So those are indicated by these colors here. And after that, you assign each observation to the cluster with the nearest mean. So in this picture here, for every single like gray dot here, we calculate the distance from that point to all of the cluster centers and then assign it to the one that is closest to it. So that's what happened here. Like this was assigned to the red, these were assigned to the green, these were assigned to the blue. Next, we recalculate the means for each of the clusters by finding like the center of that cluster. So if we go back to this image here, we recalculate the means by finding the center of these 2 points, the center of these, and the center of these. And once you have those new centroids, then you do this again where you assign each point to its nearest cluster center and you iterate until you converge. So this is an example of it happening in real time. So there's 3 different clusters here. And as you can see, as you iterate through, the different points are changing based to which cluster they're assigned to based off of the distance to the different centroids which are moving as well. So in this algorithm, the choice of K is an important parameter. You tell the algorithm how many clusters you want. So I'm going to talk a little bit about how to choose this K a little bit later. Okay, so how do we run the notebook? There's a notebook to do this in ARP. And the first step is to find the neighborhoods for each cell in your data set. So the one parameter to be mindful of here is a distance limit, and we just heard Ina talk about this and had a question about it too, of how you choose this distance limit. The other thing to think about is whether or not you want to use counts or frequency when you're identifying your neighborhood. So counts is simply the number of each cell type that's surrounding the index cell. And frequency is dividing that number by the total number of cells that are in some set radius around it. So frequency, the difference between the two is that frequency does not take into account cell density. And the choice whether or not you want to use counts or frequency just depends on what you think are different neighborhoods. So for example, if we're looking at this index cell here and the black circle indicates some radius around the index cell, and we're simply to count the number of cells around the index cell, we would have, for instance, 6 T cells, and the frequency would be 100% T cells because there are no other cells surrounding it. If we compare that with this case where we have just 1 T cell in the radius, but there are no other cells surrounding it, the frequency of these two cases look the same because they're both 100% T cells, but the count is different. So the question is whether or not you think these are two different neighborhoods or not. Do you want to pick out, very dense areas of your tissue and have them be separate from very sparse areas, even if the composition is similar, or do you want those to just be considered as the same neighborhood? So it's really up to the question that you're asking. Okay, so how to choose an optimal K? So our notebook has a few quantitative measures that algorithmically, I practiced the word last night, help you choose how many clusters you want. And so the first of these is inertia. And so inertia is calculated by measuring the distance between every single data point, so in this case, each cell and its cluster center, and squaring this and adding across the entire cluster. So you can imagine a lower inertia is better because you want all your data points to be really close to its cluster center. And if that is the case, then the number would be low. So this is a graph of what inertia looks like. So what we typically do is we'll increase the number of K, so the number of neighborhoods we're trying to find in our data, and calculate this metric for all of these different cluster numbers. And we're looking for what we call an elbow point of where it kind of like levels off. So in this case, it would be 3. So like 3 seems like it's the best choice in this situation. The second metric that we use is the silhouette score. So silhouette score is calculated by using the intra-cluster distance and comparing it to the inter-cluster distance. So the distance between like the points within a cluster and then the distance between different clusters. And then you can use this equation here to find the silhouette score. So a higher silhouette score is better. And so in this case here, maybe the best silhouette score would be like 4. So one of the downsides of silhouette score is that it's very computationally intensive. It takes a very long time. So even if in our data sets with just like 10,000, 100,000 cells, this takes a very long time, and subsampling, we would need to subsample the number of cells to make this calculation run at all. And of course, we're also doing this calculation across many different Ks, so it takes even longer. And so while like in the best case scenario, by looking at these graphs, we would be able to like find the perfect K. We'd be like, oh, it's obvious it's this one. That also lines up nicely with like the silhouette score. That's usually not the case. So in our real data, it might look something like this, where like if you look at the inertia graph, there's no obvious elbow point. And if you look at the silhouette score graph, it's like looks kind of noisy. And also a lot of times they don't even really agree. So it can be hard to figure out how many neighborhoods are actually in your data. And so we found that actually looking at the overlays and the outputs can help in making this choice. So similar to what I was talking about before with like the pixel and the cell clustering, like iterating through this is helpful. So this is another example of a lymph node. So we could see it's a different lymph node this time, but it's like still similar structure, T cell zone, B cell zone, germinal center. And so this is the neighborhood overlay where each color corresponds to one of these neighborhoods here. So what's nice right off the bat is that you can see structure. And if we like zoom into like specific clusters, for instance, this like salmon and this green cluster, they're defined by like B cells and FDCs, which we would expect because this is the B cell zone. And so if you have some idea of what neighborhoods are in your data, then looking at these heat maps and these overlays can be a good way of helping you choose your K. Like if you're seeing a bunch of things squished together, maybe you want to increase the K. If you're not seeing enough definition, you want to okay, those things I said were the same, I think, but you can it can help you choose how many clusters you may see or you may want to see in your data. Okay, so in addition to the overlays, the other heat map outputs that you get out of this notebook are first a heat map showing you the average composition of each of the clusters. So when you're actually doing the clustering, the feature vector that you're feeding into it is the number of cells in each index cell's neighborhood. So this is like an average of that. So like what if all the cells in cluster 1, what is surrounding those cells? What is composed? What is the neighborhood of the cells in cluster 1? The second heat map output is the number of each cell assigned to the neighborhood. So in cluster neighborhood 8, for instance, what are the cells that are actually in that cluster? And finally, you can look at the mean marker expression per neighborhood. So Ina talked about this a little bit, of how you can use these neighborhoods to find cool biology by looking at differential marker expression within each neighborhood. Okay, so finally, an example of this in the study of the transition from ductal carcinoma in situ to invasive breast cancer. So I talked about the study yesterday when I was talking about pixel clustering. So in this data set, we were able to identify 10 different cell neighborhoods using k-means clustering. And so if we were to just look at these next to the actual MIBI overlays, we can see it matches up well. So for instance, in this inset here, the yellow cluster is the B cell enriched neighborhood. And if we look at the CD20 signal, that matches up well with that. So yeah, so in this data set, we were able to find 10 different neighborhoods using this relatively simple algorithm of k-means. So in summary, neighborhood analysis identifies groups of cells that are commonly co-occurring in your data set, and it's a simple interpreted method for identifying your neighborhoods. Yeah, so I'll take any questions.
Questions to populate online.
Mhm.
Yeah.
I have always a question. So the k-means clustering and the other clustering method is the spatial earlier. It both need like some number of like clusters that use. But there are also clustering algorithms. Like for these ones, you need the number of clusters and then the variance of the clusters. But there are other clustering algorithms where you just assume a fixed variance and then it will find like the number.
No, we have
Okay,
not.
so
but
the
that sounds-
question was,
okay.
all of the
forget.
clustering algorithms that we're talking about here, you pick the number of clusters, and then it decides what variance difference between cells in order to fit that. You could alternatively say, this is how separate I want the clusters to be, and then it would determine how many you need to fit their criteria. Have you tried any of those?
To my knowledge, we haven't, but that's a great idea. I feel like that's something that we could try pretty easily. Yeah.
The one thing I'll say is that, like, I think this is kind of almost, I think this is very true also with like high-dimensional single cell data. Sean can speak a lot to this, is that the biggest problem, I think, with any of these clustering algorithms, whether it's for neighborhoods, whether it's for like cell types, is that I haven't seen any that with no supervision, like accurately routinely identify like low prevalence events very well. And so I think that's why we eventually end up always keep coming back and reverting to, because it's one of these things, right, where it's like, if it's not going to be so good that you can just assume that it's right, and you're going to have to go back and do that anyway, that it's sort of, this has been one of those where I don't know if we found a convincing enough way to pick a K to where we would just trust it out, like without having to check it.
Yes.
Another way to put it is, if you want to start a fight at a computational biology meeting, just ask people how they choose the number of clusters.
Right.
That's all you need to do. And so I guess what Candace and Mike are saying is that, like, you're better off being more granular and then using your expert knowledge or at least interest to drive how the last few kind of grouping steps are done. And this is not specific to MIBI or even spatial biology. This is like anyone doing single cell workflows, exactly like I said, will tell you this. Your rare, potentially interesting cell populations will always get sucked into another cluster if you try
to automatically get to the right answer without any kind of meta interventional steps along the way.
Yeah, so there's a question online that basically asks exactly what we just talked about. You know, what's the best way to optimally choose the number, the K, and what is there other approaches besides silhouette score? And the consensus from the experts in the room is that we have yet to find an approach in MIBI data, in CyTOF data, single-cell RNA sequencing data, where you can reliably just trust the output of the automated clustering. And so given that that's the case, it's unsatisfying, but it's more practical to just pick the number based on manual expert inspection right from the beginning, rather than do some hope that it would get it right and then end up changing it anyway. Okay, awesome. Thank you, Candace. All right, next up, we have Adam, who will be talking about spatial LDA.
Okie dokie. Can everyone hear me okay?
Yep, the slides look good too.
Awesome. All right. Well, hi, everyone. I'm Adam. I hope.
Just talk a little louder.
Sure. Hi, everyone. I'm Adam. I hope that, first of all, I hope that my Simpsonian depiction from Mike's slide on day one lived up to expectations. Very important that was accurate. Today I'm going to be talking to you about spatial LDA, another way of identifying microenvironments in your cell phenotype maps. All right, quick outline for this talk. We're going to ask what questions this helps us answer in particular, like why would you use this over the k-means clustering or the k-means neighborhood identification. We're going to go over the algorithm itself, high-level overview. We're not going to get too much into the nitty-gritty of like optimization steps or anything like that. We'll do a really quick notebook walkthrough, same as the past two talks. And I'll give an example application where this was more useful than, well, where you would want to, an example of where you'd want to apply this over k-means. All right. So the real meat of it is that we want to identify microenvironments. So same goal as k-means neighborhood analysis, but in k-means, you essentially, once you picked out, essentially your cells can't share microenvironments. You only get one. They're in the neighborhood, and that neighborhood is the microenvironment. So what happens if we get rid of that assumption? What if we want to see smooth transitions between different microenvironments? How would we even be able to look at that is the question. So to get some insight, we're going to borrow from the topic of language. We're going to translate concepts from linguical analysis into this sort of tissue analysis. So in language, our like quanta of information is a word, while here that correlates to a cell phenotype. A collection of words make up a topic. So things like sports, your words are going to be like a topic of sports. Your words are going to be like baseball, run, goal, stuff like that. So in the tissue analogy, that's going to look like a microenvironment. It's going to be the different cell phenotypes comprising that neighborhood. And a document is going to be the neighborhood itself. So because essentially a document is going to have a topic and a neighborhood is going to have a microenvironment. That's the analogy you got to work with here. A more visual picture of this can be seen here with some anti-cat bias that I'm upset about, but a graphic's a graphic. And here you can see that documents can actually have multiple topics, which makes sense. Flying cars are driven by cats. That's about tech and animals. And these topics are essentially defined by their word preference. And these words can also share topic preference as well. And our goal essentially is to construct this representation from just whatever set of documents we're given. We want to construct our topics given whatever words we have in there in these documents. So a tool in natural language processing that's really good at doing this is called latent Dirichlet analysis. First and foremost, it's a bag of words model, which means that the words and how close they are to each other does not matter. They're just all put into the same bag, drawn at random, essentially. And what this does is it does, as I said before, it solves for what it naturally constructs these topics, and it assigns for each word in a document the probability that it's for that topic, essentially. So walking through what this looks like more in detail, you essentially have a topic preference for each neighborhood. So that's that theta. So in this one here, it's slightly leaned towards the purple topic more than the yellow topic, and then so on and so forth. Moving along, there's a topic preference for each word in each neighborhood, which is this Z, and this will eventually be what we categorize, how we get this like probability distribution of different topics for each cell. This is our metric of interest, essentially. But moving forward, we also have a word preference for each topic K. So essentially, going back to that previous graphic, this would be animals prefer cat and dog more so than like a word like run. And then we have W here, which essentially represents our, this also represents the word, but this is the actual word in the document. So since that's the word in the document, that's our known information. And essentially using our known information, we can back propagate this to update all of these probability distributions. This is classic Bayesian inference, and you can do this with elbow method. I think that's how it's implemented on the back end. So basically, because you know what the cell phenotypes are, you can retroactively create the topic preferences from this. But shifting this to cells, the bag of cell model, we want our cells to, when two cells are close to each other, we'd really like them to have similar topics. If they don't have similar topics, then you don't get this smooth change in microenvironments. You could have microenvironments just clumped up next to each other, same as k-means. There'd be no solid advantage. So spatial LDA adds this spatial regularization term, which basically makes sure that these cells share the same topics when they're close together. And just to rehash what the microenvironment representation looks like in k-means, basically each cell inherits its neighborhood's classification. So it can only have one classification. It can't be smoothly transitioning between two. While in spatial LDA, the cell has a probability distribution of different microenvironments it could constitute. So cells in this region are mostly large, they prefer microenvironment 4 here, but there's a little bit of transitioning from microenvironment 3, a little bit of microenvironment 1, and that opens up a lot of possibilities in terms of further downstream analysis. Hyperparameters for this model are pretty much exactly the same as k-means. You're going to need a number of topics, which is analogous to a number of clusters, and you're going to need a neighborhood radius, analogous to the neighborhood radius in k-means. Walking through the notebooks, there's first the pre-processing notebook, which will help you configure these hyperparameters and reformat your data into a format that the algorithm prefers. And then there's the training and inference notebook, which will actually train and run the spatial LDA model. In the pre-processing notebook, you'll notice I'm also going to by the way, I'm skipping over the data path setup because once you're this far into ARC analysis, you're tired of data paths 100%. You know how to do it. So getting back on track, the featurized cell table, you'll notice this here. You're pretty much going to always want to choose cluster as your featurization. Markers and average marker and counts are also supported, but at this point, you're definitely going to have cell clusters, and using markers is and thresholding based on whether or not they're positive enough is essentially a manually curated way of clustering your data. So if you have clusters, we recommend that you use those anyways. And you can also, so you set the featurization there and you set the neighborhood radius there, and you'll get that adjacency plot from earlier that you'll be able to see whether or not that looks reasonable to you in terms of neighborhoods. But again, this is going to be physically dependent on your tissue type. As for computing the number of topics, this will be, since you can run k-means in a pretty quick amount of time, and you might already have that data by the time you run spatial LDA, the number of topics will be similar to that number, maybe within maybe 2 to 3 higher. So you'll have a much finer region probably than this 3 to 20 number of topics. Not to mention that we rarely ever find that there's a need to go above 12 for number of topics. So bear that in mind. The metrics for determining this are all the same as k-means. So even if you didn't run k-means, we have all the same tools for discovering the right number of topics in the notebook. Then training and inference, really the only thing other than data paths that you'll need to change here is the number of topics. The number of topics which you determined in the previous notebook. Everything else is automatically loaded in from the pre-processing notebook. So you're good to go essentially. But it should be noted that this does take a while to run, so have more productive things lined up while this runs. Depending on system, this is an order of magnitude of like adding 10 fields of view is about adding 30 minutes of runtime to the training and inference. So be aware of that. I can't really give specifics. I would say like, and I think this metric was essentially on an 8-core machine with 8 gigabytes of RAM, but hard pressed to say it. It's still accurate to an order of magnitude. And then a quick vignette of where this has been, a case where this was super useful in as opposed to k-means neighborhood identification was in tumor immune boundary investigations and triple negative breast cancer. So previously there was immune microenvironment and tumor microenvironment, and investigating the boundary between these was pretty difficult because anytime you ran k-means, it would just spit out, here's your immune and here's your tumor. And then there's the boundary between them. So essentially, if you wanted to look at that behavior and see what cells were close to that boundary, you'd have to compare the distributions of the cell types on each side of it. So I think it was like a Wilcox rank score sum. I think that was the metric that was used, but anything that compares the two distributions to see if there's a significant difference would be used here. But that's a lot of manual curation. You have to identify the boundary. You have to have your set microenvironments, your like big ones. And spatial LDA, thankfully, essentially automates that whole process because it's really good at identifying the smooth transition between microenvironments. So it identifies subtypes in the tumors that track really well with the boundary, shown here in the spatial LDA paper. And it goes further than that, and it identifies novel tumor substructures. Particularly this yellow topic 4, was even found to correlate really well with negative survival rates in TNBC. So there's an argument that it can outperform k-means in that case, especially with these sort of fine substructure cases and boundary transition cases. But to summarize spatial LDA, it's an advanced method for robustly identifying microenvironments, and it also identifies smooth changes of microenvironments at the cost of being pretty computationally expensive. Yeah, any questions? Noah, could you mind helping field questions for me?
Yes, no problem. So first question in the chat is, what do you think about running spatial LDA on whole slide images? And I would say it just won't work at all. There's a couple reasons. One, there's a lot of stuff in it. The main reason though is that spatial LDA scales non-linearly with the number of cells in each image. So it's not the same running 50 images of size 1 and 1 image of size 50. It's going to be much worse running a big one. And so that's what you get with whole slide imaging. So sorry. One thing that, as soon as Barbara's talk ended, we came up to her like, let's try this out. So the spatial dimensionality reduction she talked about had a lot of the same benefits of this, where you have a smooth interpolation of different classifications. And so that is something that is scalable. And so if we try that out and it looks promising, it will definitely get put into ARC, imported into ARC so that it can be easily used. But yeah, out of luck if you want to do spatial LDA for that in its current form.
You can always subsample. You could chop it up.
Because Adam, once you have the LDs determined, is it pretty easy to add them to fields that were withheld from the original analysis?
So is it fast to, even if you've trained a model on a small subset of the data, can you then quickly run it on large subsets of data?
Inference takes around half as long, so still an improvement, but
Still not great.
yeah, still not great. To be honest, I'm confused why there's a separation between training and inference if they're going to take the same amount of time, but that's how it's set up right now.
Would you say LDA is ideal or better for analyzing the tumor immune boundary compared to other methods, or is this just an example that they used in that paper?
This is an example they used. I mean, it's a good question. I would probably say, saying something better is purely better is a big commitment. I'll say that it was easier to run the spatial LDA to get a result about what was going on the boundary, on the boundary. But different analyses are for different questions. So in terms of broadly identifying whether or not there is something happening at the boundary, spatial LDA is great. If there's something that's really particular that you are really questioning about, then you might even want to look at like context-dependent pairwise enrichment, if it's like, pairwise behavior that you're looking for. But in this case, the spatial LDA, I think, was easier and showed essentially the same information as the previous method.
Awesome. Thanks, Adam.
Thank you.